and it is translated on the ribosomes. At that time, there are several components that are needed. So it uses messenger RNA, it uses transfer RNA, and it uses ribosomal RNA as well. So all the three main RNAs that we talked about are all utilized at the time of translation. Ribosomal RNA is a part of the ribosomes, which are the those are basically ribonucleoproteins that make the structure of ribosomes. Messenger RNA is the genetic code. This is where the codons are. Transfer RNA is the anticodon. So it consists of nucleotides which serve as anticodons, and it also carries the amino acids. So that's the amino acid carrier. mRNA is the carrier of the codon, genetic code itself. And ribosomal RNA is the part of the machinery that makes the proteins, that reads the code on messenger RNA and uses that to incorporate amino acids. This is, and in case of prokaryotes, eukaryotes, there are some differences, and differences are not in the process. Differences are in the overall or in the arrangement of the process of RNA. Both of them carry out translation the same way. So things start, then they become longer, and then at the end polypeptides are synthesized. Ribosomes, since they don't have any organelles, so all of their process takes place inside their cytoplasm. Eukaryotes are a little bit complicated in that regard. They have more structure than prokaryotes, so their level is slightly different. It is compartmentalized, so some things happen inside the nucleus, the rest of the things happen inside the cytoplasm. RNA molecules are synthesized inside, and translation, formation of the proteins take place inside the cytoplasm. So there is definitely some transport issues that are related to the translation of messenger RNA. So the translation process is polymerization of amino acids, making a longer chains, just joining the individual building blocks of proteins, which are amino acids, and making a polypeptide chain based on that. Polypeptide proteins sometimes are regarded as a polypeptide, but the polypeptide is generally just the sequence itself, not the structure. So polypeptide chain of amino acids generally mean the amino acid sequence itself, not really the folding part of the protein, because as we know, folding is important for the functioning of the proteins. When proteins do not fold properly, then they are not functional proteins. So two things which are important are ribosomes that contain ribosomal RNA, and ribosomal RNA is actually catalytic. A part of that is catalytic, and as you know, those enzymes or those molecules that serve as enzymes like RNA, those are called as ribozymes. Ribozymes are like, RNA-based enzymes. And so RNA does serve, some of the RNA molecules do serve as enzymes, so they are called as ribozyme. So in ribosomes, the catalytic activity, or at least a part of that, comes from the RNA molecules and not the proteins. The joining of amino acids to synthesize polypeptide is a role that is played by RNA, not by protein. So it's actually very interesting structure. Ribosomes are very interesting structures. The entire ribosomes have been completely characterized. Their structure has been understood and has been published now. Transfer RNA molecules, which are the carriers of the amino acids. So they carry the anticodon. Anticodon is responsible for incorporating amino acids. Amino acids are added and then it is carried at the time of translation. They are the carriers of amino acids. Ribosomes, they have an important role that they play. Both prokaryotes and eukaryotes have their own ribosomes. Organelles also have their own ribosomes as well because they carry out some translation, like chloroplasts and mitochondria have their own ribosomes as well. In eukaryotes, structurally they are slightly different. There are differences between prokaryotes and eukaryotes, and the difference is not uh, in the nature of the proteins or the RNA molecules. It's the general difference is the size and the number of RNA molecules that make those ribosomes. Oops. So these are the differences here. So ribosomes are in two parts. They have small subunits and they have the large subunits. So large subunit in case of prokaryotes is called 50S and the small subunit is called 30S. Collectively, when they are joined together, it makes a structure which is called as 70S. And this S is the Svedberg constant, which is the unit that describes describe the sedimentation. So it is the sedimentation which is not based on only the size but also the spread of the molecule as well. So that's why 50 plus 30 does not add up to 80. So it makes 70S because it is when they are together, their sedimentation, meaning that as they settle down under centrifugation, is somewhat different when they are separate. So they are probably more spread out. When it is a single structure, it is more compact structure. So that's why it's not 80S. In case of eukaryotes, it is 80S, but it consists of 60S and 40S. Small subunit is 40S and the large subunit is 60S. And both of those consist of multiple, several ribosomal RNAs as well as proteins. So the breakdown is here at the bottom. So prokaryotes consist of 23S, ribosomal RNA 16S, ribosomal RNA, and then they have the third one, which is 5S ribosomal RNA. And they consist of a number of proteins, 30 proteins here, 33 proteins here, and 21 proteins in the small subunit. In case of prokaryotes, they have 4 ribosomal RNAs. They have 28S, which corresponds to 23S. Then they have 5.8S and 5S, and then they have something similar as 16S, which is called 18S ribosomal RNA. Small subunit has as many as proteins as the large subunit of prokaryotes, 33, and large subunit has more proteins than the large subunit of prokaryotes, which is 47. So number of proteins and number of ribosomal RNA molecules are part of the ribosomes, which are ribonucleoproteins. So they consist of RNA as well as proteins. rDNA, which transcribes ribosomal RNA, those genes are located in tandem. They are clusters. There are a number of those. From last chapter that we covered, we learned that ribosomal RNAs are repeats. They are repeated multiple times, and they are part of the repetitive type of DNA that makes a big portion of the DNA. So they are not just single copies; they are multiple copies because the role that they play to synthesize ribosomes. So multiple copies are needed. They are part of the tandem repeats, which are separated by some spacers in between. So they are transcribed together, but after they are transcribed, they are broken down into smaller pieces to make their own ribosomal RNA. Transfer RNAs are generally smaller. They are one of the smaller molecules, about 75 to 90 nucleotides long, and they are transcribed from a larger precursor as well, and then goes through modifications. Modifications make changes in the bases, in the nitrogenous bases that they carry, and the overall structure that they make is a clover leaf-like structure. And they have very unusual type of nucleotides. That's not how they are synthesized. They are modified. They go through a process of modification, editing. So what we learned, editing is one of the modifications that are done on transfer RNAs or in RNAs in general. So editing changes the nature of the nucleotides or the sequences that RNAs are made of. So these are some of these nucleotides which are contained in transfer RNA. We don't see them in other types of RNAs. Inosinic acid, not found in anywhere else. Pseudouridylic acid, that is also not seen anywhere else. So they have resemblance with other nucleotides, other ribonucleotides, but they are not found in any other ones. So whenever you see their sequences in RNA molecules, they are written there, which makes them look different. So for example, here you will see this. This is the cloverleaf model of RNA, transfer RNA. You will see it has a stem, it's a stem loop-like structure. Here there are several hairpins that you can see there. So it has this D stem, and then it has an anticodon loop. So, and then it has a variable loop, and then this one has kind of a thymidine or methylthymidine like uridylic acid and C stem, and then they also have all these structures where they are in the form of helix. So they are in the form of a double helix here, but then the loops are not paired. The areas of the stems are paired. They become double-stranded there, but otherwise these areas, they are without any pairing in the loops. Anticodon is located at its base. This is where three nucleotides define what is carried on their 3' end. So anticodon, in the anticodon stem or anticodon loop, defines what is carried on the 3' end. Amino acid is carried on the 3' end, and that sequence is CCA. So CCA is one of the modifications that are done on transfer RNA. It is added after it is synthesized. So this is one of the modifications. Another modification on the RNA molecules is done on the 5', where a G nucleotide is generally the first nucleotide. No, it has no significance. Maybe the only significance is it stabilizes it. So G nucleotides phosphorylated. We've seen that in case of messenger RNA. There is also a methyl guanosine nucleotide that is added on the 5' to protect the end of the messenger RNA as well. Sometimes there are 2 or 3 of those G nucleotides there just to protect them from degradation by enzymes. So this is the general structure of a transfer RNA, and the general structure shows that the anticodons are the ones which are responsible for what is carried on their 3' end. Okay, so that's something we have already discussed. They're three-dimensional molecules. So cloverleaf model is a true two-dimensional structure of the transfer RNA molecule. The three-dimensional structure is in the form of a kind of a letter L, somewhat letter L, and that just shows something very similar to what we have seen in case of the cloverleaf model. So all of those stems and loops are here as well. So this is the anticodon loop, um, which has the anticodon on the base of that. So this is the 3' end, this is the 5' end, and it looks like kind of an inverted letter L. All the other stem loops that we have seen in case of cloverleaf model are there as well. Um, so this is what is utilized at the time of RNA molecule. The way RNA molecules, transfer RNA molecules, are charged with amino acids. They are synthesized as RNA and then they are added with amino acids. That's called charging of amino acids. It is done with the help of enzymes which are called as amino acyl tRNA synthetase. Synthetases are those that are responsible for adding amino acids to the RNA molecules. Then they become transfer RNA molecules. There are at least 20 different synthetases that are responsible for adding amino acids to about 30 to 40 different transfer RNA molecules. There are multiple RNA molecules, transfer RNA molecules that carry the same amino acid. So when they carry the same amino acid, the synthetase that is used to add those amino acids to transfer RNAs are the same. So each synthetase is specific for one amino acid. And there are at least 20, if not more. So amino acids initially, so this process of adding amino acids to transfer RNA is a two-step process. Amino acids are converted initially to an active form with the addition of ATP. So ATP energy is utilized, ATP gets hydrolyzed, and it produces a molecule which is called amino acyl adenylic acid, and that adenylic acid is utilized in the second step to be added to transfer RNA. So first amino acid is activated, and then in the second step, that activated amino acid is added to transfer RNA. So this enzyme is highly specific, and multiple transfer RNA molecules are served basically by the same synthetase that is called an iso-accepting transfer RNase. It accepts more than one transfer RNA to carry out the same function. So these are the two steps that are shown here. So in the first step, synthetase, this is the amino acyl synthetase, the enzyme that it utilizes ATP to activate the amino acid. So amino acid, which is shown here, amino acid added with the energy of ATP, it gets phosphorylated, activated, phosphorylated, which is shown here. So you see amino acid along with a single phosphate here. Two of the phosphate groups are detached from the triphosphate molecule. And then in the second step, that activated amino acid is added to the 3' end of the transfer RNA. Phosphate group is removed from them or utilized, and as is hydrolyzed, energy is required. So that energy is provided by ATP. So two-step process. The first step is in which the amino acid is activated with ATP, and in the second step, that activated amino acid is added to transfer RNA. Okay, any questions? Feel free to interrupt. Whenever you have any questions, just stop me wherever you need any help. Just let me know before I go to the next step. So the second part is related to the 3 steps of translation. So 3 steps of translation. There are 2 videos uh, in the